Good afternoon or good morning, as the case may be. Hey guys, and welcome to your second lesson of the week, topic 8.1, metabolism and competitive inhibition. This will finish up topic 8 for us and allow us to start preparing for our next topic, which will be topic 3, genetics. And this is really getting down to the very end of the year. We're going to finish up topic 3 and then topic 10, which is a very short topic, and we will be good to go, having gotten almost all, except for one, of the core topics done, and several of the advanced topics, meaning next year we can focus on some of the more advanced topics as we get along in IB biology. Don't worry, they're actually quite fun. Some of them are in getting into the very end, so pretty exciting. But let's go ahead and dive into metabolism and competitive inhibition. Okay, so thus far we've talked about two big processes in topic 8.1. We've talked about photosynthesis and cell respiration, and both processes were the conversion of energy from one form to another in endergonic and exergonic reactions to be able to transform energy into a form that could be useful to us on the dailies, right? So we took sunlight, which is not inherently useful to us except for some of the heat that it might provide our bodies to keep us warm. And it converted that mostly uses energy of sunlight into chemical energy of sugar and photosynthesis, which was then converted in respiration from the chemical unrefined energy in sugar to the refined chemical energy in ATP after respiration. And there we go. We were able to power our bodies with that energy and we're good to go. So now in topic 8.1, we're going to talk a little bit more in depth about how these enzymatic reactions are actually performed, go into a bit more depth about how that energy is conserved and different ways that these reactions can be cut on or off. We sort of explored it a little bit when we talked about the production of leptin to reduce levels of appetite and therefore respiration. We talked about the shutting off of photosystem 2 during cyclic photophosphorylation to make sure that we had enough NADP plus being regenerated so we could run that process. But today we're going to talk a little bit about we could about how we could directly affect the enzymes and change their functionality as well. So let's dive right in. So the first thing to understand is that when I use the word metabolism in this particular lecture, I will be referring to any reactions that help maintain homeostasis. Normally, I would use metabolism as a common term to describe cell respiration. But in this case, I'm going to go ahead and use it to describe any reactions within the body that are actively promoting the chemical reactions we need to survive. So the first thing to understand about any metabolic line is that it happens in small steps as opposed to one big one. It is really important that we do not try to do the very complex things that are occurring within our body in one step. And the reason why is that these reactions actually take an enormous amount of energy. Now, because we split it off into small sections, you take it one little bit at a time, you don't really notice, right? And it's maintainable, it's manageable. But if you were to do any of the reactions in your body all at once, you would create so much heat and you would require so much energy that you would probably spontaneously combust. You would definitely shut down. It would not be able to work. I think I used an analogy for this earlier when I said that if you're cleaning your house and you're doing a deep clean, right? You don't just do the deep clean all in one step, right? You're not just like, I will clean every room in my house all at once, and you split into 16 different people and you do that. That would take an enormous metabolic cost. If you could imagine spending the energy you spend, scrubbing the toilets, vacuuming the carpet, washing the carpet, sweeping the floors, mopping the floors, washing the dishes, whatever you do during a deep clean, you can imagine doing all of that stuff all at the same exact instant. That would take so much energy. But instead, to make it manageable, it might take the whole day, but we do it in small steps. We clean the kitchen first, then we clean the bathroom, then we clean the living room, then we clean the bedroom, whatever. We go through some sort of step-by-step process to allow for that energy expenditure to be a little bit more regulated. We also, in doing so, regulate the quality of the reactions. So, as you're probably aware, when we multitask, oftentimes we sort of let things slip. The quality of the work can slip as we multitask, but, if you don't multitask, if you focus on one thing at a time, you often produce things of better quality. Now, you may disagree, but the data shows that for most people, that's how it goes. Now, that means that when we are performing metabolism, really what we're trying to do is we're trying to sequester the various different parts of this process into smaller chunks that allow us to make what we want to make and only make exactly the right amount of what we want to make, no more and no less, so we can conserve energy. All metabolic reactions happen in two ways, either a cycle or a chain. A chain is just where we go from point A to point B in one direction. It is non-cyclic, it just keeps on going. A cycle is when we have a regenerative molecule, so something that we always start back with, and we might add things in from an external chain, but the cycle itself continues basically the same as it always does and makes the same product out of the same reactants. So what are some examples of cycles we've seen? I want you to sort of brainstorm that for a second. You can pause the video if you'd like. Okay, so now that you've had a second or two to think, let's go ahead and think of some examples of cycles we've seen. The two big ones that we've talked about thus far are the Calvin cycle, otherwise called the light independent or a misnomer, dark reaction. This happens in the stroma of plants, and its goal is to go ahead and fix CO2 in its atmospheric form into RUBP, ribulose biphosphate, to create the intermediate molecules that will eventually be used to produce glucose and to regenerate RUBP. So it's a cycle because it always comes back to that RUBP. The other cycle we learned about was the Krebs cycle. This happens in the matrix of the mitochondria, and the Krebs cycle starts with acetyl-CoA, which is the product of splitting a couple of carbons and other molecules off of pyruvate, which themselves were the product of splitting glucose. So we have acetyl-CoA, and acetyl-CoA goes around and around in a circle, and it helps to reduce NAD and FAD into NADH and FADH2. And it also helps to phosphorylate some ATP, so it makes ATP out of ADP. And that cycle comes back to acetyl-CoA. And the chain that leads to it, of course, is glycolysis, right? So glycolysis produces two molecules of pyruvate. The pyruvate is going ahead and transformed into the acetyl-CoA during the link reaction, which is the little reaction that occurs just before the Krebs cycle. Some of the hydrogens and oxygens and phosphates are popped on and off, and we end up releasing CO2 and then going through the Krebs cycle. So two big cycles and lots of chains. Both electron transport chains are example of chains. Lots of chains we've learned about. Okay, so how do enzymes play into this? Enzymes are the mediators of all metabolic activity in the body. When a metabolic reaction has to occur, an enzyme is there. Enzymes help to reduce the amount of activation energy necessary for a reaction to occur. So if you guys look at the graph there, you can see that without the enzyme, this reaction would have needed a lot of activation energy. With the enzyme, it did not need much at all. And usually the differences between the enzyme with or without sort of tell you how effective that enzyme is as a catalyst, right? Which means just a mediator of the reaction, right? So are you a good mediator? Well, if you are a good mediator, then you probably make a huge difference in the amount of activation energy needed. You probably reduce it quite a bit. But if you're not a particularly good mediator or catalyst for a reaction, then you probably don't reduce it all that much, right? So if we saw a greater difference between the two curves on the graph to the right, then we would say that it was probably a good mediator. And if we saw less of a difference, we would say probably that it was not a great mediator. So a lot of the energy for your reactions in your body is conserved by enzymes. Much of the energy for activation energy is actually supplied by the heat from your body, but it's not enough. If your body temperature was the right temperature to actually sustain all of the reactions in your body without enzymes, you would probably be on fire. You would be like some sort of crazy magma being. But we can't be that, so we need enzymes. Anyway, how do enzymes work? So we kind of talked about this previously, but just to go back, enzymes are strings of amino acids that have folded due to the chemical properties of their R groups into various different conformations. These conformations have made small spaces in the three-dimensional structure of the protein or enzyme, which can accept outside molecules. We call this little pocket that molecules can bind to the active site, and we call the molecules that bind into that pocket the substrate. And we can see here that a little substrate here, that little red and green thing, is going into the active site of this enzyme. The enzyme does its reaction, and it splits the little substrate into two products. So whatever comes out of the enzyme, whether it's being put together or taken apart, are known as the products. So basically, enzymes function just by bringing substrates closer together. Literally, that's all it is. They just bring them closer together and kind of, kind of push them together, almost like, you two should kiss. And then, they either go together their separate ways or whatever happens. But basically, the enzyme is just the mediator again for this to occur. Okay, so the lock and key versus induced fit. So there's two different models that we've come up with to sort of describe how substrates bind to enzymes and how this happens. The first one, the older one, is the lock and key model, which you can see at the very top. The lock and key model just basically says that the active site is basically the exact same shape as the substrate, and this helps to make sure that each individual enzyme is only interacting with its particular substrate. Some enzymes can be more general, but most enzymes are extremely specific, and this is another way of your body to help control which reactions happen when and where. So if the right enzyme is not available, the reaction can't occur. If the right enzyme is available, only that enzyme can make that reaction occur, right? And this helps your body just to regulate stuff. So the old model was that the enzyme accomplished this specificity by having an active site that only matched the substrate, and then the substrate would bind, and then the enzyme would do its thing. The new model, the induced fit model, is the more accurate run, and this is produced by scientists, having a better molecular idea of what's happening. You can see that the enzyme's active sites are not at all, or they might barely resemble the substrate's shape. When the substrate binds, though, the active site of the enzyme changes as the enzyme's conformation changes, and it actually has a tighter, snugger fit into the substrate. So notice that when you look up at the lock and key model, there's some space and weird little spaces between the substrate and the enzyme. But in the induced fit model, there's not. So it's a perfect fit. And this is how enzymes retain their specificity. It does not, however, mean that that enzyme could just glom onto any substrate. It still has specificity of one way or another. Okay, so an enzyme can do two basic functions. When we talk about metabolism, of course, we're talking about carrying out metabolic functions. What does that mean? It means we're splitting molecules apart or we're putting them back together. So we have special names for this because, of course, we do. Metabolism is when we take small molecules and join them into larger molecules. This would be like when we are using our condensation reactions, otherwise known as our dehydration reactions, to create a large macromolecule out of its monomers. For example, to go ahead and create a long molecule of starch or cellulose from our glucose monomers, or for another example, to create an enzyme or protein out of a stream of amino acids using peptide bonds. That's anabolism. And enzymes help that to occur. They also help catabolism. So catabolism is to break bonds apart. I don't have a great way of remembering anabolism, but I do have a great way of remembering catabolism because cats are like mobile blenders, and they tear everything apart. And so when I think about tearing something apart, I think of cats, and when I think of cats, I think of catabolism. You are welcome. So catabolism is just to break those bonds in order to make these smaller molecules. I apologize if some weed eater audio gets into this video. They are weed eating the yard outside of my house and also leaf blowing it, so this is going to be interesting. Anyway, so anabolism typically tends to be endergonic. It takes in a lot of energy. So think about anabolism happens a lot in photosynthesis when we have to take the sun's energy and combine it with water and carbon and all that stuff in order to create glucose. So we build a large molecule out of smaller ones. So anabolism is usually, again, endergonic, whereas catabolism usually releases more energy than it takes in. So catabolism would be more like respiration, where we break apart the large sugar into smaller pieces in order to help to phosphorylate ATP. So here we can see, exergonic reaction, free energy, that would be more like our catabolism. Endergonic reaction would be more like our anabolism. And we can see that with the enzyme, it takes less energy both times with the enzyme than without to make this occur. Okay, so now, how could enzymes be directly affected to change metabolic rate? This is, we talked about this before when we talked about denaturation, but denaturation is not the only way that you can affect an enzyme's rate of reaction. In fact, there's two very common ways that we can do this, which is called inhibition. So there are molecules called inhibitors. To inhibit, of course, means to slow down or to stop something. And inhibitors go and they do just that. They bind onto enzymes and they help to prevent their functioning. And inhibitors, you might think, well, that's totally a bad thing because enzymes are good. Well, but your body doesn't always want your enzymes to be doing their thing, because there are times when your body just doesn't need what the enzyme produces. How does it cut it off? Well, it could destroy the enzyme by denaturing it, but if it did that, it wouldn't be able to use the enzyme later. So how do you cut it off without having to break it? I mean, if you want to think about this in an easy kind of way, you know, when you are done with your television set, hopefully you don't just stand up and you pick up a sledgehammer and you go, well, I'm done watching this. Right? Hopefully you cut it off with the remote. So you can think of the remote as being the inhibitor. The remote is inhibiting your television from showing you stuff even though it didn't destroy it. You can always come back and press the button on the remote again to cut your TV back on. Okay, so there's two basic kinds of inhibitors. The first is called competitive and the second is non-competitive. Competitive inhibitors are going to act as much like substrates as they can. So basically, they're going to pretend to be the right kind of substrate. And they're going to go in and be like, oh, hello, would you like to bind to me? And the enzyme is going to be like, oh, yes, I would love to bind to you. And the enzyme binds to the substrate, to the competitive inhibitor, and oh, the real substrate comes by and is like, oh, John, I can't believe you're going out with that competitive inhibitor. And the enzyme is like, gosh, I didn't know. But regardless, the substrate is unable to bind to the enzyme, and the reaction rate is severely slowed or stopped. And then there is the non-competitive inhibitors. The non-competitive inhibitors are going to bind to a secret site called the allosteric site. It's not the active site, it's this whole other site on the enzyme. The non-competitive or allosteric inhibitor is going to bind to that allosteric site. And when it does so, it actually changes the conformation of the active site so the substrate can't bind. So the substrate is like, oh, my love, my one and only, let me bind. Hello? Anybody there? There's no active site for them to bind to, or it can't actually get their shape. So non-competitive inhibitors or allosteric inhibitors are the more powerful of the two. And we're going to take a look at that in a second. But if you kind of think about it, it makes sense. The competitive inhibitors are still going to get competition from the substrate. But once the allosteric inhibitors bind, there's no chance for the substrate to bind. So one of them, the competitive inhibitor, is going to have a weaker effect than the allosteric. So here we can see the competitive inhibition, and this thing, this diagram is really great, but at the end it says there is no reaction. At that moment, there's no reaction, but if you kind of think about it, there's a bunch of enzymes floating around in one place. Some of them are going to have the actual substrate bind versus the competitive inhibitor. So the competitive inhibitor might be able to take down one enzyme's functionality, but not many others. Usually it just slows the reaction. But here you can see the enzyme, the substrate and the inhibitor look very alike. The inhibitor comes in, beats the substrate to the goal, and blocks the active site. Here's our non-competitive inhibition. So again, we have that that allosteric inhibitor binds to the active site and changes its conformation so that even if the substrate is present, there's no way it can bind. So here are some inhibitors that IB wants you to know. I have blocked out whether they are competitive or non-competitive. And so they actually do want you to know these. So they want you to know the dihydropteroate synthetase. I don't even know what that does. It says it binds reversibly to the enzyme's active site. I don't even, anyway. Its substrate is para-aminobenzoate, and the inhibitor is sulfenzidine. And we can probably assume that because it says under the binding section that it binds reversibly to the enzyme's active site, and while it remains bound, substrates cannot bind. If it's binding to the enzyme's active site, what kind of inhibitor must it be? So I'll give you a couple of seconds. You can pause the video if you like. Correct. It's a competitive inhibitor because it is binding to the active site of the enzyme and competing with the substrate, the real substrate, para-aminobenzoate. And then there's phosphofructokinase. This one is a fun word, but in this one it binds to a site away from the active site, which must be an allosteric site, so it must be a, you guessed it, allosteric inhibitor, and changes the active site conformation. We'll actually talk about that second one here in a minute. Okay, so competitive versus non-competitive. If you had to look at this graph and determine which one was competitive and non-competitive, Which one would you choose? Well, they're already labeled. Wow, that was an oversight on my part. So I'll just make a quick comment that what I wanted you to notice about this before I ruined the experience was that the competitive inhibitor in this case is doing far less to actually reduce the rate of reaction. You can see the rate of reaction still reaches the maximum rate of reaction, and it's still going up fairly quickly, though less quickly than with a normal enzyme. The non-competitive inhibitor, on the other hand, notice how much it really inhibits this reaction. It's going to stop it because there's no way that even if the substrate is present that's supposed to bind, there's no way it can. So if you really want to stop a reaction, use a non-competitive inhibitor. So here's a good example of negative allosterism. So this just means allosteric inhibition. So phosphofructokinase, PFK, which we just learned about, is involved in glycolysis. Yes, you remember that process. So actually, PFK is involved in the breakdown of glucose into pyruvate, but when ATP binds to it, it actually stops it from splitting glucose. So in this case, ATP acts as an allosteric inhibitor, which is kind of funny because normally you think, oh, it's got to be something else. No, it can be any molecule that binds into the allosteric site of an enzyme. Now, why does this happen? Okay, so imagine that you're running a crap ton of cell respiration and you have tons of ATP. In fact, you have more ATP than you know what to do with. Your body's like, ATP, you get an ATP, and you get an ATP. And everybody's like, wow, I actually have too much ATP. Maybe I should slow down the respiration so I don't just keep wasting energy making more energy that I don't need. And so when there's a crap ton of ATP just floating around, some of it is going to start binding to PFK if there's enough. And if there's enough to bind to PFK, you do not need to worry about producing ATP. So it actually stops PFK from performing glycolysis and thus slows down or stops respiration, which is really cool. Of course, when the ATP is needed, that's bound to PFK. That ATP will leave PFK and go do its job someplace else. And when it does so, it allows PFK to start its job again, which is probably means that more ATP is needed and that respiration is needed. So it's a cool little way of controlling the speed of that chemical process. But however, there can also be positive allosterism. So allosteric molecules are not always inhibitive, and neither are allosteric sites always inhibitive. In fact, in hemoglobin, which is a molecule, it's a protein that carries oxygen through your bloodstream, hemoglobin is actually activated by its allosteric site. So when oxygen binds to its allosteric site, it actually makes the hemoglobin more willing to bind to more oxygen, which is a good thing because it's an oxygen carrier. So the hemoglobin is induced to bind to more oxygen, and boom, you get this chain reaction where the hemoglobin starts picking up a lot. Now, when the hemoglobin is induced to release its oxygen, like when it gets to an area of the body that needs it, when it releases the oxygen in surrounding tissue, when the oxygen leaves the hemoglobin, it actually makes it more susceptible or less susceptible to holding oxygen, and it's able to offload easily. So it's pretty cool how allosteric sites can change those substrates, those active sites, to make them more or less willing to bind onto different molecules. Now, the last way of inhibiting a molecule is through end product inhibition. So end product inhibition is going to be when the product at the end of a long chain of metabolic activities actually ends up being the inhibitor. So while you're making the stuff you want to make, you end up making the inhibitor as well. So we can see this is actually the chain that IB wants you to know on the right-hand side. It's not really all that difficult. You just need to know that you start with your initial substrate, which is threonine. This is an amino acid, which you could probably tell by the onine at the end, just like methionine or I can't think of any other glycine. Anyway, so threonine is going to go ahead and bind into the active site of an enzyme known as threonine deaminase, and the threonine deaminase is going to guide it through one of those long chains we talked about. You don't really need to know anything about the enzymes in that chain, just know that they're slowly breaking down threonine into a different product. The end product is isoleucine. Oh, well, there we go. There's another amino acid right there. So it turns it into isoleucine. And isoleucine is interesting because isoleucine can then go back and bind to the exact enzyme that takes in threonine and inhibit its active site. Why in God's name would you want this to happen? Okay, well, say you need a lot of isoleucine. Say your body's making proteins that require a lot of isoleucine. Well, you can produce a lot of isoleucine from threonine, which might be more abundant. But as you're doing so, eventually you'll get to the point where you produce more isoleucine than you need. So the extra isoleucine is going to be hanging around near the enzyme. And there it goes, just like the ATP with the PFK, it goes ahead and it binds onto that enzyme and it makes makes it so that the enzyme stops the process and doesn't make more isoleucine when it doesn't need to. Why can't we just use a switch to cut this stuff off? Well, it's not how your body evolved. I'm sorry to tell you, but your body just doesn't have an electrical system. It can just be like, oh, and so it's got to figure out how to do this the hard way. So it uses end product inhibition. End product inhibition, if you want to think of a daily example of end product inhibition that you interact with, is your toilet. So, which is like, what? I don't know if you guys have ever done this. You lift the back off the toilet and you flush. There's like a bunch of water in the back. And the water in the back is actually pushing up on this little float. If you don't know what I'm talking about, go look at your toilet after this to see what I'm talking about and flush it so you can see. So when you flush it, all that water in the back of the toilet drains out and it goes into your toilet bowl to help it flush. And when that happens, the little float that is in the back of the toilet goes down with the water level. And as the float goes down, it actually pulls up this little plug out of the toilet. And this allows more water to run back into the back of your toilet. But as more water runs into the back of your toilet, it lifts up that float. And as the float lifts, the plug closes so that once your water level in the back of your toilet has reached the original level, it seals that little plug and no more water flows into the back of your toilet and the whole thing stops. So it's pretty cool. So take a look at how your toilet works, and that's a great example of end product inhibition. The thing that you produce, the water, actually stops the entire reaction until you kickstart it again. Okay, so how can we use the knowledge of end product inhibition or of inhibition at all to our words? How can we use inhibition of enzymes to help? That's what I'm trying to ask. So let's go ahead and take a look at a cool example of this. I'm not going to play the audio because the audio would probably screw up with the recording mic on this laptop. And also the guy's voice is awesome, but very difficult to understand. For me anyway, he has a very thick accent, which is cool, but I'm just going to go ahead and play it. So, if you guys, for those of you guys who don't know, malaria is a parasite that infects a lot of people. It is delivered via mosquitoes into the bloodstream, and it infects your red blood cells. As the parasite or plasmodium, it's a type of protist, goes into the red blood cell, it sort of injects itself inside, and it begins to develop into its next life stage, which helps to propagate it, to breed more and more. So we can see it's dividing rapidly here, and it's going to infect and take over the red blood cell, and once it does so, it's going to burst out and basically become pathogenic. So it's going to release more of itself, just like a virus, and infect other red blood cells. This makes you very sick, as you can imagine. So what's really cool about inhibitors is that once we discovered them, we discovered we could use them to help stop processes like this. So to stop inhibition like this, what we've done is we realized that in order to divide so much, they desperately needed these proteins that right here, which we can see that are spinning out these little protein fibers. These proteins are usually called centrioles, and they aid in cell division. And that cell division is the problem, right? Because if the plasmodium isn't allowed to divide within your red blood cells, it starts to take over. So what if we could inhibit that enzyme? So that's what they did. They went ahead and they figured out which inhibitor would break down this enzyme, and they inhibited it. And they made it so that when it tried to divide again, it was actually unable to do so. So we went in, here we go, here's the centriole that produces those protein fibers. And what we did is we went ahead and attached an inhibitor onto its allosteric site. It completely changed it, and it shut it down. So that's a pretty cool way that we can use inhibitors, and that leads us into the different kinds of medicine that we can do today that we couldn't dream of earlier. And this comes down to the design of drugs to help us fight off diseases. So the current model and that we've been using since we discovered inhibitors was that we figured out whatever enzyme needed to be inhibited, like that centriole, that division protein. We went ahead and we screened that enzyme against a huge database of inhibitors that we had started to gather to figure out which ones might be a potential match, just naturally. And once we chose it, we had to modify it because usually it wasn't exactly the inhibitor that would work on that enzyme, but it was close, so we had to modify it chemically to be more powerful and to fit with the molecule we wanted it to fit with. And then we would release it into the body, and voila, the process would be stopped. Malaria would be defeated. But now, oh, this is cool. Now we have this weird sci-fi thing called rational drug design. And the idea behind rational drug design is that we go ahead and we use a computer to scan the three-dimensional shape of the enzyme we want to inhibit, and then the computer designs the perfect chemical inhibitor for that molecule, and we synthesize that in the lab. I just think that is so cool, and it really speaks to me about how mechanic this whole process is, how mechanized it is. You know, it's wild that we can go in and design a molecule for this to occur. That just blows my mind. And I think that's so cool. But anyway, rational versus drug design is a great application of this, and we're going to look into that here after you're done with this. Okay, so one of the things that IB wants you to be able to do is to be able to determine how effective an enzyme is as being a chemical catalyst in a metabolic reaction. So is it, like we talked about before, does it do a good job of lowering activation energy or does it not? And we also want us to be able to calculate the rate of reaction, which we did in our enzyme lab, but I'm going to go ahead and have us sort of renew that here. So this is going to be our practice here in the very beginning. This will be some of the practice that you do, and then your extension will be to look into rational drug design because it's really super freaking cool. So let's start here with the effectiveness of enzymes. I'm going to start showing you how to do this, and then you guys are going to go ahead and finish it with the practice in some of the questions. And I'm going to show you how to do this, and then I'm going to let you guys answer these as well as part of the practice. So this will be your little practice problem stuff, and then we'll take a look at that cool inhibition stuff in the next part of the lesson. So let's go ahead and begin. So we'll start with this guy and this great database question. I'm going to move this over here into our notebook, our handy dandy notebook. Sorry about that. I'm just going to go ahead and use the scissor tool because I think it's probably going to keep rejecting me, and I don't want this to take a lot of time. Okay, so let's go ahead and take a look at this. So the degree to which enzymes increase the rate of reactions varies greatly, but if we calculate the ratio between the rate of reactions with and without the catalyst, we can determine how effective that enzyme is. So basically, we just look at it, how well did it do with and how well did it do without the enzyme. If the difference isn't a lot, then the enzyme is probably not all that necessary. If it's a huge difference, then wow, that's an important enzyme. So let's go ahead and I'll get you started, and I'll show you how to do one, and then you guys can do the rest. So the first thing is state which enzyme catalyzes a reaction with the slowest rate in the absence of the enzyme. So first we have to calculate these rates. And I'm going to show you how to calculate the rates, and then I'm going to let you guys answer these questions yourselves via the Google form attached to this. So how do we calculate rate of reaction? Well, the first thing to understand is that we're going to be looking at a specific ratio. Okay, so here in 3 it says calculate the ratios between the rate of reaction with and without. So we're always going to have the with first, and the without second. That's very important. And we can see here with and without. That order is not just it's not just random. So you need to make sure you're paying attention when you're reading problems like these to make sure which is coming first, with or without. So we're going to do the with versus the without. And to do this, we're going to need a scientific calculator. Now, I don't have a scientific calculator, so I'm going to go ahead and type it into here. So let's take a look at our problem here. Okay, so let's see how they got this one. So they say this is the ratio between how they get it. So this is the rate with enzymes, is 1.0 times 10 to the 6th power. If you guys are familiar with scientific notation, that's what this is. So I'm going to go ahead and type that in, 1.0 times 10 to the 6th power. Okay, and then we know a ratio is a division problem, so I'm going to divide that by this guy, the without, over here. So it's 1.3 times 10 to the negative 1. So I'm going to go ahead and say, so here we go, divided by 1.3 times 10 to the negative 1. Okay, now I'm just going to go ahead and press enter, and here we go. Now, if we're looking at the scientific notation here, it's coming out as this big number. If we go back over here, we can see 10 to the 6th and then 10 to the negative 1. Usually we do this as like a subtraction problem, so it would be 10 to the 5th, which is what has happened. And so when we are going to convert this guy into scientific notation, we need to take it back so that there's just one decimal place, so behind the 7. So here, I'll show you what I mean. It's easier to do it than to talk about it. So I'm going to take this decimal point, and I'm going to go 1, 2, 3, 4. Okay, so I'm going to go 1, 2, 3, 4, and that's going to give me this right here, which is 7.69 times 10 to the fourth. Okay, so over here, I'm going to end up, this is going to be, I'll make it a little bit smaller here. So this is right, 7.69 times 10 to the fourth. Okay, now we can see here that this number here, it says 10 to the sixth. We go back over here, 1, 2, 3, 4, right? So here we've gotten a different result slightly than the one that they've gotten. 10 to the sixth versus 10 to the fourth. Let's go ahead and see. It should still work though. Let's go ahead and see what we got with the other one. So 6.4 times 10 to the fourth. 6.4 times 10 to the fourth. All right, so I got that one, and then divided by 1.7 times 10 to the negative 7th. So we're going to go ahead and say 1.7 times 10 to the negative 7th. Okay, and there we go. Now we're going to go ahead and run that division problem. And here, okay, so we're going to go ahead and move this up so that it is behind the first whole number here. So we're going to go 1, 2, 3. Okay, so it's going to be 3.76 times 10 to the negative third. And the reason why is we started here, we go 1, 2, 3. Left is negative, to the right is positive. So we go 1, 2, 3. So 3.7 times 10 to the negative third. Okay. Okay, and you just go through and look at those. The biggest numbers, I'm gonna warn you about this in scientific notation. The biggest numbers are gonna be the ones with the positive values, right? The smallest numbers are gonna be the ones with the more negative values. So be careful about looking at these numbers and just assuming or deciding that these are gonna tell you which one is bigger and which one's smaller. Now in this case, 7.69 times 10 to the fourth is a greater difference. And so we can see if we're looking for here the ratios between the rate of reaction with and without an enzyme, well, in this case, 7.69 times 10 to the positive fourth, that's pretty big. So that's a big ratio between the rate with and without enzyme. And 3.7 times 10 to the negative third is pretty small. So already I can probably say that carbonic anhydrase, even though I don't know what it does, is probably far more useful as a catalyst than carosteroid isomerase. So go ahead and calculate all the ratios, select which one has, which enzyme catalyzes reaction with the slowest rate in the absence of an enzyme. So when you look over here, which one catalyzes it with the slowest rate, all right? In the absence of an enzyme, that means which one is the slowest is OMP decarboxylase, if you're wondering. You guys can, I'll just go ahead and give you that one. Okay, and then you want to look for the one at the most rapid rate. So I went ahead and chose this one because it was the slowest, but it says which enzyme catalyzes its reaction at the most rapid rate. So it's asking you with the enzyme, which one catalyzes it fastest. And interestingly enough, that's also OMP decarboxylase. Okay, now I want you to go ahead and calculate the ratios between all the rest of them and see if you can explain the differences between them based off of what we've already talked about. It should be more than possible. Sorry, I gave you 1 and 2, but I just wanted to make sure there wasn't confusion with those because that's usually where the confusion comes from. So go ahead and calculate the rest of that for your Google form, and you should be good to go. Now we'll go back to this here, and for the final little bit, this is calculating the rate of reaction. So when we look at this graph here, I'm just going to kind of lead you through it because we already did a little bit of calculating the rate of reaction when we made our respirometers. But here I'm going to go ahead and include a little bit more instruction in that. So here what we would do is Our rate of reaction, it says here, is this crazy formula. Don't worry about that. The rate of reaction is really just what you end up with. So your final amount of concentration of whatever substrate minus what you started with divided by the time. Okay, so if I go ahead and I pick up this graph right here and I move it over and I'm looking at the reaction rate of all this stuff, here it looks like it's based off the same kind of lab that we did. So it's using catalase to split down hydrogen peroxide. We'll go ahead and take a look at the reaction rate right here for this guy. It's right at this point, and actually I'm going to go ahead and use 60 because that's the nearest number that I can actually use to get all of these, because notice that there's no precise number I can use over here. So I'll use 60. So at this point, which I'm going to pretend like is my final point here, I have about 21.5. I have about 21.5% oxygen. If I look back at what I started at, back at the beginning of this reaction over here at 0, I can see that I had maybe 20.2 or something like that. Okay, so 20.2. So I'm going to come back over here. I'm going to say minus 20.2. So my final, what I ended up with, minus my initial, what I started with. And then I'm going to divide all of that. I'm going to put these in parentheses so we can know what we're looking at. I'm going to divide all of that by the amount of time. And here its time is in seconds, and that was 60 seconds, so I'll divide by 60 seconds. And let's go ahead and plug that into our calculator. So 21.5 minus 20.2 equals 1.3 divided by 60 gives us 0.022. So 0.02 is what I'll say. And what are our units? 0.02. And we're going to say this is going to be 0.02% oxygen per second. So that's our reaction rate. And we can calculate the reaction rates of all the rest of these guys down here. I'll go ahead and type up the formula here for reaction rate, just so you guys can remember what it is. It is the final, and I'll say product, minus initial product, divided by your time. Okay, guys, and finishing up that problem based off of that is just going to be down here. I'd like for you guys to go ahead and take a swing at that, and that'll be your practice for today. I hope you enjoyed it, and that's it for topic 8. We'll start our review for the lessons for next week, and we'll get ready for an assessment, which will either be open note on this Google Classroom or it'll be in person. I'm still getting those resources together and still trying to decide what I want to do. So, all right, guys, enjoy.